Welcome back, everyone, to Vox Markets. My name is Paul Hill, and by popular demand, I am delighted again to be able to speak to CEO Alastair Smith and Chief Scientific Officer Fiona McLaughlin of Avacta. So welcome, guys.
Hi, Paul.
Hi, Paul.
Yeah, well, a lot's been happening since we last spoke, not least the excellent news on the dose escalation of the AVA6000 last week. But before we sort of dig into the details, Alastair, I don't suppose you could give us a quick sort of progress report on reformulating the rapid lateral flow test.
Yeah, of course, Paul. I mean, obviously extremely unfortunate that the Omicron variant affected the performance of the test. As I think we've explained, the combination of an AFMA and an antibody in that test, it's the antibody that has failed. Unfortunately, the AFMA works extremely well with the Omicron variant. So we now are searching for another AFMA or antibody to pair with the existing one, which isn't necessarily a long process. What we don't know is we don't know how the regulators will view the changes that we make. So it's very difficult for me to give you any sort of timescale because we don't know how protracted that regulatory process will be. But I guess one of the other benefits in this very commoditized market now for antigen testing is that we'll be able to do some work to get the cost of goods out of the product as well. So I can't really give you a very accurate time. As soon as we have a clear view, we will of course update the market.
And what was the actual problem with the Omicron variant? It was just they had lots of different mutations on that spike protein that made it difficult to actually sort of like identify it.
A huge number, over 30, and mutations within the nucleocapsid protein as well. So, all of these antigen tests are vulnerable to mutations, and whatever the next mutations are, could equally affect other tests on the market.
And how do you actually future-proof that going forward? Is it possible, or have you just got to be very reactive, or you just got to sort of like get the genomic sequencing data from, the relevant labs or so?
Yeah, so I mean, there probably isn't time now to go into this in detail, but because we now know a lot more about the virus and the antigens that we're interested in, and because we know the AFMA that we having the test is unaffected, that's taught us quite a lot about which epitope and which part of the antigen is least vulnerable to mutation. So we can obviously apply some thought to generating a secondary agent that gives us a bit more future proofing going forward.
Yeah. Well, one area, and just turning now to the Prodoxorubicin, the AVA6000 that has just been absolutely doing really well over the last sort of like 6 months since it started its sort of clinical trial back in August. Could you quick remind investors of the sort of what it does, the AVA6, how it actually works, and the sort of purpose of the dose escalation, which was obviously the news last week?
Yeah, sure. Well, I mean, the comment I'd like to make perhaps before handing over to Fiona is just to focus investors' minds, as we have done in the past, Paul, you and I, on where the real value drivers are for shareholders in Avacta. And it is the oncology drug development and the opportunities there that really drive significant value in the business. So I think, despite the obvious disappointment and setback and delay in antigen testing revenue, fortunately, the big value driver is really motoring extremely well. Fiona, can I hand over to you just to talk about 6000 and the phase 1 trial?
Sure. So AVA6000, as you know, is a prodrug, and it contains the active component, which is a very well known and understood anthracycline. So it inhibits the DNA synthesis and the dividing tumour cells. It binds to an enzyme called topoisomerase 2 and inhibits the activity of the enzyme. So topoisomerase, the topo comes from topology, so it changes the shape of the DNA and the ability to form that helical structure that you always see. So the molecule itself, doxorubicin, is incredibly well understood, and that's the benefit of this molecule, because once you're in the body, the way that the drug works is not novel, so we completely understand the mechanism of action. And as you know, it's going to be selective for those tumour cells which are high in FAP expression. So it's targeted. So it's the first time that there's been clinical data on a targeted doxorubicin molecule that's FAP targeted, I should say.
And what's the importance of the dose escalation? I mean, just from a layman, obviously you've now decided to go from sort of is it 80 milligrams up to 120, I think it was, from last week, which is obviously a huge 50% increase. And it does seem as though the like for doxorubicin in normal, sort of without any of that protection of the prodrug, it does seem as though that's the sort of is it between 65 and 70 or something like that is the maximum tolerated dose. So you're now heading to sort of like uncharted waters as much as anything, because Prodoxorubicin isn't allowed normally because of its safety, I guess.
That's exactly right. So the purpose of the phase 1 is to really determine the safety of the molecule, not the efficacy. So you're absolutely right. So the starting dose was just below, it's 54 milligrams of doxorubicin, so just below the dose that's used in clinic. What's actually important is the cumulative dose of doxorubicin. So the dose over time is what drives the long-term, the chronic cardiotoxicity. That's what we know and what we understand. That cumulative dose of AVA6000 at the maximum might be quite different to doxorubicin. So that's what we need to understand. So that's why, as well as plasma levels, we're measuring urine levels of the drug, but we're also, where we can, we're taking tumour biopsies, and we're going to be measuring AVA6000 and doxorubicin levels in the tumour biopsy. And that's going to be really important data when we get biopsies, of course, because patients have to consent to give them in this part of the trial.
And how have you actually been measuring safety so far?
It's standard criteria that are used in all oncology phase 1 trials. So you'll measure bloods. So you'll look at white blood cell counts, platelet counts, neutrophil counts. Of course, because it's cardiac toxicity as well, we've got various different measures of biomarkers for cardiac output. We've got scans, we've got echo scans in certain patients as well. So we're following it in very standard ways. In fact, all the criteria are listed on the clinicaltrials.gov website.
Okay. And when it comes to actually the decision by the safety panel, what have they actually decided that it's positive and it can be sort of escalated? It seems like a huge increase. Obviously, they seem to be pretty confident in the safety profile.
Well, again, the increase from one dose cohort to the next is actually driven by the regulatory authorities. So they recommend what the next dose escalation will be. So again, we're not doing anything novel here in terms of the escalation. And the safety, the SDMC, the safety committee, actually review all of the data to date. But the members of the committee are actually primarily the PIs involved in the trial. So they've actually been meeting every fortnight and reviewing the data. So it's just a coming together of everyone to make that formal decision, which is signed off.
Okay. And how many patients have actually been dosed so far?
So we don't say exactly how many patients, but it's a 3+3 trial design. So I think you can probably figure it out. So
right. Yes. Okay. And just broadly, Alastair, in terms of the timescales, where do we stand in? Is there been any sort of like impact at all from Omicron? Obviously, with all the pressures on the NHS with regards to sort of patient recruitment or anything like that, or is it still very much on track?
It's on track. There has been a little bit of delay, just as you described, because of staff in hospitals being ill with COVID and so on. So nothing unusual, and we're on track. Middle of the year, end of Q2, middle of the year for the phase 1A dose escalation that we've just been talking about. And then, assuming all is well, we'll progress on to the dose expansion phase, the phase 1B, in Q3 of this year.
And what's the purpose? You can remind investors the purpose of, is it obviously to test the tolerability, but to also find, is it sort of like a maximum tolerated dose? Is it for the 1B side of the phase 1?
Yeah, that's right, exactly that. So just as Fiona described, a standard 3+3, each cohort has 3 or so patients. The dose is escalated, as you've just seen, based on positive safety data. And that allows us to identify either a maximum tolerated dose or a recommended dose for the next phase, the dose expansion phase.
Okay. And Fiona, when is the PK data likely to be released, and what significance is it?
So we'll release the PK at the end of that first phase of the escalation. So if you bear in mind that these patients, they're all terminal stage patients. and they'll at different tumour types, so they're at different stages of disease. So you'll get more variability we would expect in the escalation than the expansion, because in the expansion, obviously we'll have one tumour type per cohort. And so you should get more consistent data. The data we have to date, because we're analysing it real time, and of course that was presented in the SDMC meeting, has actually been remarkably consistent. So we're very happy with the data that we have so far.
Okay. So in terms of sort of the actual, when do you think the PK data will be released? Did you say that was sort of?
So we haven't set a date yet, Paul, but that data will be released as part of the end of that first phase of the trial. We'll
okay.
Put all the data together.
So mid-year or something like that.
It won't be before then. Yeah. Yeah.
Okay. And then Alastair, just to sort of like obviously the confidence level in terms of anecdotally, is there any, I mean, I know it's very, very early days, but is there any anecdotal feel at all of how the sort of like the drug is performing in the tumours, etc., the efficacy, or is that just really sort of asking too much?
Well, as Fiona said, it's not really appropriate to talk about the data in the middle of the study. I mean, I've said publicly, and Fiona's just said as well, that we're very happy with the data. It's very consistent, and we're very happy with the data so far. But it is an early stage that we're at, so not appropriate to really set up any signposts yet. But very happy with what we've seen so far. Okay.
And can you just talk about the US trial equivalent of the AVA6000, and likewise with the new candidate as well, because it does sort of like give an indicative feel that certainly to investors that everything seems to be sort of like moving in the right direction.
Yeah, sure. I mean, I'm happy to comment on the US trial, which obviously hasn't started. We have the IND approved. We're now working to get two trial sites in the US up and running, and those will. So that's part of the same phase 1 study. It's not an independent or a repeat study. Those sites in the US will contribute to the phase 1A and 1B alongside the sites in the UK. And then I don't know, Fiona, do you want to comment on 3996 and the choice of a preclinical development candidate?
Yeah, I'm very happy to. So in fact, Paul, I've been the project leader for AVA3996 since I joined Avacta 12 months ago. So I've been really focused on making sure we have the right molecule to take forward. So we've been taking a number of studies over the last 12 months. We call them de-risking studies, because we already had a lot of data on the molecule, but we wanted to make sure that any risks were taken out. So we've been working with CROs throughout the UK and mainland Europe, and we're performing the standard safety efficacy studies, for example, on the molecule. And that was the data we reviewed towards the end of December and into January with a bunch of external consultants that we work with in toxicology, CMC, regulatory. And we came to the unanimous decision that there was nothing to stop us moving forward to candidate selection. And we've planned that process out. So now we're working full steam ahead to get as much data as we can, as quickly as we can.
Okay. And is this to treat sort of like is it for a multiple myeloma, is it? But it's a hard tumour again, is it?
Yeah, that's a really good question. So right now, proteasome inhibitors are really only registered to work in multiple myeloma. And that's because of a very narrow therapeutic index. So the drugs work incredibly well, but they do have effects on other cells in the body. So the idea of having a targeted proteasome inhibitor opens up not just myeloma, but other tumours in which you might have a high FAP expression.
Right.
And so that's what we're working on preclinically, is to get that data to support the hypothesis that it might work not just in hematological tumours, blood tumours, but also in solid tumours.
Okay. And then Alastair, sort of like bigger picture wise, assuming that the actual phase 1 trial goes well, so that at the completion of sort of like phase 1A and phase 1B, and we have obviously all the safety profile and the tolerability and the maximum dose levels, plus maybe a sort of like a very early sort of bit of information in terms of whether the efficacy, etc. What's that mean for the company in terms of sort of like the big broad picture with lots of different candidates in oncology and also the precision platform?
Yeah, well, I think that's a really important point, that what we are evaluating here in this phase 1 study is not just specifically AVA6000, a prodrug of doxorubicin, but we're evaluating whether the precision platform, in simple terms, works in humans in the same way that we've seen it in animal models. So, the PK data and perhaps a hint of distribution and efficacy that we see from the study, as Fiona's described it, in the middle of the year roughly, is a huge value inflection point, because it will tell us in some detail how the precision platform is working in this first in human study. So, the potential value for the company is enormous. It's a massive milestone for us. It tells us that the pipeline of potential precision prodrugs, of which 3996 that Fiona was just talking about, is the next one. But there's quite a significant pipeline of drugs that we've talked about before, and we've certainly hinted at in presentations before, that will be unlocked if we see really positive data from this phase 1 study. So it's a major value driver in the middle of this year, Paul.
Okay, and that's the end of is that the end of the phase 1A? So is it rather than just the phase 1B? So, because I think the phase 1B finishes at mid 2023, doesn't it?
That's right. So we will get a lot more information about the performance of the precision platform in human in this phase 1A study. Now, clearly, there is a lot more work to then do with the 6000 itself, but we will get a lot of information about precision in the middle of the year this year.
Right. Okay. And then, in terms of actual funding, what would be the options available to the company for sort of like phase 2 or further expansion of the precision platform to other candidates?
Well, there's the broad range of options that I'm sure everyone can figure out with with the sort of clinical data that we hope we'll see in the middle of the year. There, of course, is potential to form a license deal, a commercial deal around 6000, and potentially the precision platform. We know that we can generate greatest shareholder value by taking some of these assets forwards ourselves into the clinic, and so that's a priority for us. And we're in a strong position, thankfully, good balance sheets, and we're able to deliver the milestones we've talked about with 6000. So there's plenty of options, but we're not looking to raise funds in the near future at all.
Yeah. And then Fiona, just finally, in terms of any sort of like risks that obviously the scientific board will be looking for. I mean, does it, with the FAP enzyme, for instance, does it sort of also sort of like is FAP, the enzyme, also in patients with other conditions at all, like, I don't know, arthritis or Crohn's or other stuff like that? You'd have to wean them out, or what are the risks associated with it?
Yeah, you're right. I mean, any site of active fibroblast remodeling, you'll get some transient FAP activity. It's not clear that FAP activity remains for very long. So, for example, if you had recent surgery, if it was a breast cancer patient, for example, then there have been reports of FAP. It's certainly FAP expression, but expression isn't the same as enzymatic activity.
Right.
Okay, so you might detect the FAP there with a molecule that binds to it, but you're not actually necessarily looking at activity. So what we're actually doing in the trial is we're taking patient plasma, and we're going to be measuring FAP activity in the plasma, because it's reported that up to 20% of that FAP might not be active. Right.
Okay. And so if you did have any patients or a cohort that you felt as though you'd tested before you actually gave them the actual drugs, so it was like a personalised medicine, actually, I guess it could be, to make sure there's no vulnerable patients out there at that time.
Absolutely. So one of the things that we're doing in the trial is we're taking biopsies and also archival biopsies, so biopsies that could have been taken a year or so before the patient comes on trial. And we're working up methods to stain, to look for, again, the expression of FAP, because it's not necessarily the case that plasma FAP will correlate with tumour FAP. And that's one of the pieces that we want to try to put together. Right.
Okay, then finally, sort of, Alastair, just on the news flow going forward, I guess the prelims, are they coming out in April? Are they? Anything else sort of like,
Yeah, early April, late March. I haven't fixed a date yet, but that's the rough timing. news flow, I mean, the real value drivers, as we said up top, was around the oncology drug development. So 6,000, big value inflection point, as we said, during the middle of the year, and progress into phase 1B. The progress of 3996, we'll keep the market updated with that, that molecule as well, and aiming for an IND filing next year. And then there's our partner programs with LG Chem and Daewoong Afiexel, which are both progressing extremely well. So there's a lot to come this year, Paul.
Brilliant. Well, look forward to hearing that. You've certainly whet my appetite. And thanks again for your excellent comments, guys. And very much looking forward to see how the progress continues. So thanks very much.
Thank
Pleasure.
you.
Thanks, Paul.